Hi, it's Violet Johnston from BIOL220H. I'm going to go over the three slides that we weren't able to earlier this week. Unfortunately, I was pretty sick and I still sound a bit congested, but I'm getting better. So that being said, let's go ahead and begin. If you have any questions, you can shoot them my way, otherwise towards Professor Bloom. So on the canvas, you should have access to the slides and you'll have access to this video. So let's go ahead and start. The first thing that we're going to talk about is metagenomics. So I'll give you a moment to pull up that slide. Okay. And then we're going to talk about the Human Microbiome Project, and lastly, we're going to go into transcriptome analysis. Okay, so firstly for this slide number 1, so it's the one titled Metagenomics. Basically, it discusses on how environmental genomics, it's the study of genetic material recovered directly from environmental samples. So this includes just like looking at dirt or even seawater or even air. And instead of isolating and like culturing individual microbes in the lab, this is when researchers will extract the DNA from the sample at once. So you have an actual sample of dirt in which you're extracting DNA from. And this is really important in different applicable reasons because a lot of microbes that you find in nature, they cannot be grown in traditional lab techniques. So you're going to have to take these environmental samples. So once you have this environmental sample, what do you do next? Well, you then use a method called whole genome shotgun sequencing. We've talked about it before briefly in class, and I'll talk about it a little bit more here. And basically, this is a process that involves like randomly breaking up the DNA into smaller fragments and then sequencing them. And then we're basically able to assemble the sequences and then identify which organisms, so which microbes the DNA came from. And then we're also able to see like what genes and then what functions are present. So after that, when it comes to more of the applicable reasons for this technique, it's more so like looking at the microbial communities in like the given environment. I don't know how much you guys have heard about, like, we've been doing a lot of research on different saltwater environments and the bacteria and microbes that thrive there. I forget the name, but it's like underwater volcanoes where you can't find other live organisms except for a lot of bacteria. And we're discovering more and more every day. So it's very, very interesting. And yeah, basically metagenomics helps us understand like how these microbes interact in their environment and what role they may play in like ecosystems. And how it can change with shifts in pollution and climate change. Okay, so that's kind of the overall explanation. Let's look at the figure. So the figure there, it's going to be two pie charts. And what these two pie charts are comparing are the types of organisms represented in different genomic databases. So if you look at chart A, the data comes from like publicly available genome databases. And it looks at how the majority of predicted proteins in these databases come from eukaryotes. So it looks like 63% roughly. And on the other hand, though, for this pie chart, it says that bacteria only accounts for like 28%, while viruses and archaea make up much smaller portions, around 7 and 2%, respectively. And this, what do these numbers mean? Well, basically, you have an overwhelming number of eukaryotic data, and it shows how these public databases are very biased towards showing eukaryotic organisms. And this is likely because they're easier to find, they're easier to sequence in labs. You don't have to go out and get like an environmental sample. You can culture them in labs pretty easily. But if you look at chart B, so this is the second pie chart, on the other hand, it talks about data that comes from the Global Ocean Sampling Project. So I talked earlier about how there's like really interesting environments with underwater hot volcanoes with a lot of bacteria. So this is looking at the different oceanic environments. And it uses environmental metagenomics to sample organisms directly from these ocean environments. And in this data set, it says the majority of predicted proteins, around 90%, are from bacteria, and only small portions of the predicted proteins like from these environmental and ocean environments are from eukaryotes. It's less than 3% that is actually predicted to be in these ocean environments. And so now we see these massive differences in numbers between these two pie charts. And we're seeing that we are basically excluding data from the equation, and we have to consider environmental metagenomics, because if we don't, we're missing out on this large collection of data. This large possible collection of data about microbes and bacteria that we didn't even know played such a big role. So it basically, all in all, what I want you to get from this is how environmental metagenomics allows us to see a different picture of life, showing that like bacteria dominates the natural environments like the ocean. Okay. As well as like how chart A shows how it there's underrepresentation of microbial diversity compared to like the metagenomic studies, which is shown in chart B, which shows like a much larger bacterial presence in the environment. Okay, next, scroll to slide 2. So this one's a little bit brief. This is the Human Microbiome Project. So we talked about the Human Genome Project. With that being said, the Human Genome Project sparked a lot of different ideas for diving a little bit deeper into its ultimate goals. For the Human Microbiome Project, it studies the collection of microbes that live in and on the human body. A very popular one is talking about the microbes that live within your gut, so within your colon, but microbes are not found just within your colon. They're actually found everywhere, including on your skin too. With that being said, because they're found all over the body, they're much more numerous than people realize. And we find out that microbial cells actually make up about 1 to 3% of a person's body weight. And there's an estimated 38 trillion microbial cells in the human body. Now, take that with a grain of salt. It's just an estimation. There could easily be more or less. And I also want you to consider how did they find that number of 38 trillion microbial cells? They used to believe that for every microbial cell, there would be 10 human cells. But actually, after further research, they found out that there's about 1.3 microbial cells for every human cell. So there's even more microbial cells than we originally thought. Okay. Moving on to slide 3, let's look at transcriptome analysis. So we've talked about various techniques throughout this class for different purposes. And before transcriptome analysis and like microarray analysis, we used northern blotting. So we already discussed that, but we're going to go further into microarray analysis. Okay. So transcriptome analysis is the technique that's used to study genes which are actively expressed within a cell or tissue at a given time. And although like all the cells in an organism generally contain like the same DNA, the different genes are turned on or turned off depending on the cell type and conditions. We learned this in like our basic molecular bio classes about how gene expression fluctuates from cell to cell. So this means that like gene expression can vary between tissues. And this will also depend on like responses to factors that may happen with disease or environmental stimuli. So let's talk about microarray analysis. This is one particular method for transcriptome analysis. In this approach, it's when you take thousands of known DNA sequences, you fix them onto a glass slide in tiny spots, and each represents a specific gene, and then you can take RNA from cells, convert it into like complementary DNA. You may want to label it with like a fluorescent dye, and then you apply it onto the microarray slide, and it'll bind to the matching DNA sequences. So then you're able to see like the fluorescent signals if you dyed it, and you're able to see like which genes are expressed and then how strongly they're expressed. So you're looking at which genes are turned on and which ones are not, and if they're turned on, by how much. So with the development of microarray analysis, we now have researchers who are able to analyze the expression of thousands of genes at once, which is huge for advancing research. And again, like before we had this, we had to just rely on northern blotting, which is good and has its own merits, but it's slower and it's not as comprehensive. Like you're not able to study as many genes with northern blotting. I don't know if you guys have done northern blotting before, but usually you have to only use like a few genes at a time. And then I don't talk about it here, but if you were to go like further beyond microarray analysis, you could talk about RNA sequencing. We did briefly discuss this in class as well. It's been brought up a couple times. RNA sequencing says it's said to be more accurate. I don't know the whole truth there, but it can detect unknown genes and like RNA variants. And then it also provides like more detailed information about the gene expression, so which genes are turned on. But yes, so that hopefully only took about 10 minutes of your time. These 3 slides are very important, so I'm glad I got to cover them. And yeah, thank you so much, and thank you, Professor Bloom, for giving me the opportunity to do this for you guys. Have a wonderful day.